I'm really excited now to introduce our next speaker, Sylke Poehling, who is the CEO of Spark Therapeutics, SVP at Roche. Also has a long history of thinking about working in the space of genetic medicine. She's both experienced with the science, but also with the business. And so to help us understand what we can expect in the future and to give us optimism for how we can get there, Sylke, please.
Well, good afternoon. And first of all, I want to thank you, Eric, for organizing this wonderful conference. And the morning sessions have already been great and really led to a lot of inspiring conversations and ideas. So today is about genetic agency. And when I reflected about the talk, I of course thought, what is genetic agency and how can we create the conditions for genetic agency? Right. And the way that I thought about it, actually it came to my mind that maybe we need to actually look at the genetic medicine age as the past, the present, and the future, and really define 3 eras of genetic medicine. And then identify how can we actually create a path, how can we build bridges between the different eras. So I believe firmly that we have a mandate. And the mandate is a scientific mandate, but most importantly, there's a mandate by patients who are actually looking to us to develop transformative medicines. And genetic medicines can be transformative. But I think now the mandate is to actually ensure that we are bringing genetic medicines to more patients and create population scale impact. What you see on this slide is actually the different eras as I define them. Era 1 has already demonstrated that we can impact the lives of many patients by our genetic medicines. And the first medicines were the medicines in rare monogenic disorders, where we had a curative intent. Now we are moving towards Era 2. There we are trying to actually bring genetic medicines to common diseases, to polygenic diseases. And mostly what we are trying to do is actually modifying the diseases. But I think the true potential of genetic medicines at some point will actually lie in Era 3, where we can precisely modulate diseased cells and turn them into healthy cells. And this is, I think, the promise of gene therapy, and how do we get there? All this work in Era 1 has been actually work of pioneers. Scientists, doctors, and most of all, patients who were hopeful and also courageous enough to actually pioneer those treatments at great personal risk. So how can we make sure that we create this population scale impact? I don't think that going from one era to the next is a linear process, or that it's just a scale problem. I think there are some very fundamental differences and fundamental barriers to moving from the first era to the second era and the third era. But before I go into that, I just wanted to have this slide as inspiration because it shows the successes that we have already created with the genetic medicines of Era 1. And we can be very proud of what has been achieved already. However, those therapies also really reminded us and enlightened us on what the limits are that we need to overcome. And when I talk about the fundamental problems, I'm not talking about what I call the table stakes for AAV gene therapy. And these are problems that we all know very well, and we are already trying to solve them. And it's fundamentally not important whether we are tackling rare or common disorders. These problems, these table stakes, need to be on our radar, and we need to actually improve in all those aspects. So when I talk about the fundamental problems, I talk about really barriers to bringing gene therapy to prevalent and polygenic disorders. And I called them the burden of certainty, the justification hurdle, a complexity barrier, and the delivery dilemma. And I will go into a bit more detail in the next slides. So when we talk about gene therapy, usually what we're talking about is a therapy that has the advantage of being a permanent therapy. But there lies also the problem for the polygenic common diseases. Irreversibility means you can have no doubts about your target and your biology. Right? You need to have absolute certainty, and we know that this is really a hard call. Causal biology is hard, and we also know that for most of our treatments in pharma industry, we are actually failing because of a lack of efficacy in most cases. Right? We are actually not that sure about target biology and what to do exactly. So mandating causal biology, being absolutely certain, is important if we want to actually treat patients with genetic medicines. And what we are currently doing at the brink of era 2 is we are looking for targets that either have a high genetic validation or that are so-called consensus target, that everyone would agree are targets that we need to tackle, or we are looking to other modalities and we are looking to validation through other modalities. So this is fine as a beachhead approach, but the question is, where do we go from there? The justification hurdle to some extent is related because it actually is the question of what's the evidence bar that we actually need to clear when we want to go into the prevalent common diseases, into era 2 and era 3. What kind of effect size do we need? What kind of treatment burden is acceptable? And these are important questions because if we don't actually make gene therapies as safe as other treatments, these are questions that will be asked, right? Do I have an effect size that creates this kind of benefit risk that actually I buy into as a patient and as a stakeholder community? So the call is actually justification hurdle, a durable and high-value treatment. On the right-hand side, we are talking about the economic challenges of bringing gene therapies to the masses. And I actually agree with Eric, who said in his podcast, his recent interview, that actually is quite good if we are focusing on the common diseases, if we are trying to, get to the economies of scale, if we're trying to overcome those problems, this will actually serve all patients. Right? If we actually make gene therapies very accessible, very safe, then the rare and ultra-rare populations will also benefit from this. So, hurdle 3, the complexity barrier. When we look at what we are currently trying to do in era 2, it is very similar still to what we did in era 1. We are selecting targets where we believe that actually modulating this one target will have a big effect size. Downregulation, upregulation, replacement. But as I said, the true hope for gene therapy and for genetic medicines is a different one. That you can modulate complex networks, that you can actually do something more sophisticated. And we know that synthetic biology generally could achieve that. We see this already in ex vivo cell therapy, but we are far away still, I think, in the in vivo gene therapies. So how do we get there? And then we have a delivery dilemma, as we all know. Right? We know what our wish list looks like. We know that we want to treat all patients, we want to redose with the same gene therapy, but also maybe with a different gene therapy. Gene therapy should not be I have to be careful. If I choose this one, I can never again get a genetic medicine. So the wish list is clear, and I think we are fighting the good fight, but we are fighting the good fight against a very formidable enemy, and that is human biology. Human biology doesn't want us to deliver genetic payloads to everywhere in the body. And this is true for AAV-based gene therapy, but also for LNP-based non-viral gene therapy, and you have overlapping problems, but you also have very distinct problems. So if we could fix this problem, I think we could already do so much more with gene therapy. So 4 major profound barriers. What is actually the solution? I believe that there's only a path forward if we actually really deliver on what I would call a convergence. Convergence of technology and biology. The path forward is actually really not an evolution from where we are, but it is a revolution. It's a revolution on the payload side to get more precise and more controllable. It's a revolution on the delivery side, where we just need to generate more optionality to deliver very diverse payloads safely and repeatedly. And for biology, we can hope that omics, AI, human model systems will actually give us much more confidence in which targets to actually interfere with and how. But what we also need to do is we need to make sure that we have a better translational understanding of our gene therapies. Catastrophic events, like we have seen again and again, are just not acceptable. We need to understand our therapies better. We need to have a better mechanistic understanding. My problem that I fell in love with is actually genetic medicines for brain diseases. And I think this is a very good example of why a revolution is needed. What you can see in this illustration, without any need to read it, is that brain diseases demonstrate to us what is so difficult. It is not just delivering something to the brain and then you can fix Alzheimer's or Parkinson's disease. The needs for the individual diseases are so different. We need to distribute to different regions in the brain, we need to deliver to different cells, and we need to actually really deliver different types of payloads to actually solve all those problems. So I think this is an important lens, and why it's very daunting, it's on the other hand also really important to look at what we are doing through the lens of the target product profiles that we need in the individual diseases, because that can actually help us to be laser sharp in our focus on the problems we need to solve. I think the good news is that for the payloads, we have already made quite some advances. We have moved beyond gene augmentation. We have so many more options nowadays. And we also have made strides on gene regulation and restricting or fine-tuning our gene therapies, our payloads. But I think we can acknowledge that, honestly, the payload revolution has actually really outpaced our ability to deliver those payloads to the necessary cells and tissues. So again, delivery is the limiting factor. And I think also for the brain diseases, I think I agree with something you said, Eric, and that is AAVs are terrible, but they're still the only workhorse we have if we think about brain diseases. There's really nothing I see out there that could replace them. So we actually need to work with them, and we need to make them much better. And that is what we set out to do, right? We know what we want to optimize, but I think we also know that even there, we need more optionality. Creating blood-brain barrier crossing capsids is important, but we also need different options. Because what you can see here is that we will still have different problems. We will have therapies that need to be delivered locally to just one brain region, and you can do that maybe by a more invasive procedure or maybe by focused ultrasound. And you will need those therapies that you can actually really deliver to the brain and that distribute across the entire brain. So still a lot to do, and we need to complement those efforts actually with a better mechanistic understanding of the capsids that we are creating for each of those cases. So this brings me to the biology revolution. We talked about biology revolution in the sense of getting more confidence in targets, but I also actually want to understand so much more about what our AAVs are actually doing in the body. We are talking about liver detargeting, but what does that actually mean? Do they actually still get into the cells or not? We use one big word, but it can mean so many different things. We don't really fully understand blood-brain barrier crossing for capsids. We don't really know exactly what happens in transcytosis versus transduction for the different capsids that we have created, and that is a huge gap that we need to close. And I think the best way to close it is to really sever our link to this over-reliance on non-human primates and invest into better human disease models. We are doing that at Roche together with our Institute of Human Biology, and we hope that we can create much more meaningful models to actually understand exactly what our major workhorse is doing in the different cells and when it crosses the blood-brain barrier, to be able to actually predict catastrophic events and make sure they don't happen. So those are the revolutions, and I think you can probably agree that there's still a lot to do. But you can also see that in many of those areas we already made quite some significant strides. So where are we now? I think the mandate is clear. We need to actually move from era 1 to era 2, and hopefully we can actually achieve era 3. But it requires actually a collective effort from everyone in this room and our network. We need to actually really focus on the big problems that we need to solve to be able to create the conditions for genetic agency. Right? We already have transformed patients' lives, and we are at the brink of delivering gene therapies to more prevalent diseases, to more patients who can benefit from this wonderful way of actually transforming patients' life. So I want to close with this slide, which shows the entrance of the Spark offices and the motto that Spark created at some point: we don't follow footsteps, we create the path. And I think that is exactly the type of pioneering spirit that we will need if we want to create the conditions for genetic agency. Thank you.